Leucine's new role, how a common amino acid powers mitochondrial energy. A groundbreaking study published in Nature Chemical Biology reveals that the amino acid leucine, long known for its role in muscle growth, also plays a direct and powerful role in regulating mitochondrial energy production. This discovery opens new avenues for understanding metabolism, aging, and neurodegenerative diseases. Leucine as a metabolic signal. Mitochondria are the cell's energy factories, converting nutrients into ATP. While leucine has traditionally been viewed as a building block for proteins, researchers from the University of Cologne and the University of Pittsburgh found that it also acts as a metabolic signal that enhances mitochondrial function. Leucine activates glutamate dehydrogenase, GDH, an enzyme that helps convert amino acids into energy. This activation leads to a boost in ATP production, especially in energy-demanding cells like neurons. The process is fast and direct. Leucine doesn't just support energy through protein synthesis, it triggers it. Implications for brain health and aging. The study focused on neurons, which are highly sensitive to energy fluctuations. Leucine's ability to stabilize mitochondrial proteins and enhance energy output could have major implications for neurodegenerative diseases such as Alzheimer's and Parkinson's. Age-related mitochondrial decline, which affects muscle and brain function. Metabolic disorders, where nutrient processing is impaired. Lead author Xiaochu Li emphasized that leucine's signaling role offers a new perspective on how nutrients can directly influence cellular energy balance. A new frontier in nutrient science. This research reframes leucine as more than just a supplement for athletes. It's a regulator of mitochondrial dynamics, capable of influencing how cells respond to stress and energy demands. The study used advanced imaging and biochemical techniques to track mitochondrial changes in real time. Leucine was shown to prevent degradation of mitochondrial surface proteins, helping maintain energy output under stress. Where to find leucine? Leucine is abundant in high-protein foods. Eggs, meat and poultry, dairy products, soybeans and legumes. While supplements exist, the study suggests that dietary sources may be sufficient for most people, though clinical applications may require targeted dosing. Click-code-seq uncovers DNA strand biases in oxidation and depurination. A pioneering study published in Nature Chemical Biology introduces a powerful technique called Click-code-seq, which reveals previously hidden patterns in DNA damage across the human genome. This method exposes how oxidation and depurination, two common forms of DNA damage, occur with distinct biases between the two DNA strands, offering new insights into aging, cancer, and cellular stress responses. What is Click-code-seq? Click-code-seq is a click chemistry-aided sequencing method designed to map DNA modifications with single nucleotide precision. Developed by researchers from ETH Zurich, the University of Zurich, and Technische Universität Dresden, this upgraded technique allows scientists to track oxidized guanines, 8-oxo-G, a marker of oxidative stress, apurinic sites, where DNA bases are lost due to damage. By applying Click-code-seq to human cells, the team discovered that these modifications are not evenly distributed. They show strand-specific biases linked to transcription and chromatin structure. Why it matters. DNA damage is a key driver of aging, neurodegeneration, carcinogenesis, chemotherapy drug action. Understanding where and how damage occurs helps researchers pinpoint vulnerable genomic regions and develop targeted therapies. The strand biases revealed by Click-code-seq suggest that transcribed DNA strands are more protected, while the non-transcribed strands accumulate more damage, a pattern that could influence mutation rates and disease progression. Key findings. Transcription-linked bias, oxidation and depurination are more frequent on the non-template strand of actively transcribed genes. Chromatin influence. DNA wrapped in nucleosomes shows different damage patterns than exposed regions. Repair dynamics. The study hints at differential repair efficiency between strands, possibly explaining mutation hotspots. Lead author Vakil Takaviv noted that Click-code-seq provides a high-resolution map of DNA damage, revealing how transcription and chromatin shape the landscape of genomic vulnerability. Future applications. This technique could revolutionize how we study cancer genomics by identifying mutation-prone regions, aging research through mapping age-related DNA damage, drug development by assessing how treatments affect DNA integrity. It also opens doors for personalized medicine, where individual DNA damage profiles could guide therapy choices. Ancient lichens rewrite Earth's history. A new discovery by paleobiologists is prompting scientists to rethink the early stages of life on our planet. Ancient lichens, uncovered in fossil deposits in North America and Greenland, may represent some of the first complex life forms to colonize land. Why it matters. Lichens are symbiotic organisms combining a fungus with an alga or cyanobacterium. Known for their resilience, they can survive on bare rock or in Arctic deserts. The discovery of fossilized lichens over 1 billion years old suggests that life established itself on land far earlier than previously believed. The new findings. Researchers used modern isotopic analysis and advanced microscopy to confirm the biological origin of the remains. The structure of these ancient lichens indicates they may have played a key role in early soil formation and in triggering Earth's carbon cycle. Implications for science. These findings change our understanding of the planet's ecological history. If lichens existed so early, other terrestrial ecosystems might have developed sooner than expected. This insight also improves our understanding of how life adapted to new environments and prepared Earth for later biological booms. Conclusion. The study of ancient lichens not only adds new chapters to Earth's history but also reminds us that even the humblest organisms can be catalysts of global transformation. Hypersonic levitation, a breakthrough in single-cell cancer research. A revolutionary technique called hypersonic levitation and spinning, HLS, is transforming how scientists isolate and study individual cells, especially cancer cells. Developed by researchers at Tianjin University, this method uses acoustic waves and MEMS, microelectromechanical systems, to gently separate cells from tissue without damaging them, offering new hope for cancer research and other biomedical fields. What is hypersonic levitation and spinning? Traditional cell isolation methods often involve mechanical cutting or enzymatic digestion, which can harm fragile cells and distort their properties. HLS changes the game by using ultrasound-driven liquid jets to peel single cells away from tissue in a contact-free process. A metal probe emits billions of vibrations per second into a water enzyme mixture surrounding human tissue. These vibrations generate acoustic waves, which form liquid jets that isolate cells gently. Once separated, cells are levitated and spun in fluid, allowing researchers to view them from every angle using advanced microscopy. This technique enables rapid and minimally invasive isolation, preserving cell integrity for high-resolution analysis. Why it matters for cancer and beyond. Most cancer deaths are caused by metastasis, where a few aggressive cells spread and form new tumors. Understanding these rare cells is critical, but isolating them without damage has been a major challenge. HLS offers a solution. Isolates 90% of cells in just 15 minutes, compared to 70% in an hour using conventional methods. Preserves delicate cell structures, making it ideal for single-cell sequencing and biochemical profiling. Applicable to fields like immunology, neuroscience, stem cell biology, and infectious disease. Dr. Shuashin Duan, who led the development, described HLS as a gentle, invisible hand that operates within fluid, not directly on the cell, minimizing stress and preserving function. How it works. The system uses 3 MEMS-based resonators to vibrate tissue in a water enzyme solution. A signal at 2.49 GHz triggers a printed circuit board to send high-frequency voltage, activating an inverse piezoelectric effect. This creates acoustic waves that flow and spin the fluid, generate liquid jets to isolate cells, suspend and rotate cells for full-angle imaging. The result is a non-destructive, high-speed method for isolating rare cells. Challenges and commercialization. While promising, HLS faces hurdles. High-frequency sensitivity. Some cells may react to acoustic fields. Lab reliability. MEMS devices in fluid can drift or require calibration. Cost and accessibility. Commercialization will be key to widespread adoption. To address these, the team launched Convergency Biotech, aiming to build user-friendly HLS workstations for labs worldwide. Tiny bat, big strategy. Fringe-lipped bat hunts like a lion to maximize energy. A new study reveals that the fringe-lipped bat, Trachyps cirrhosus, a small predator from the forests of Panama, uses a hunting strategy strikingly similar to lions. Despite its tiny size, this bat captures large prey with remarkable efficiency, challenging long-held assumptions about predator behavior and energy use. Lion-like tactics in a tiny package. Researchers equipped 20 fringe-lipped bats with miniature biologging backpacks that recorded movement and sound. The data showed that these bats don't chase prey like most small predators. Instead, they use a hang-and-wait ambush strategy, relying on acute hearing to detect frog mating calls and other prey sounds. Bats remained stationary for 89% of the night, conserving energy. Hunting flights were brief, median duration just 8 seconds. Success rate was 50%, far higher than lions, 14%, or polar bears, 2%. This strategy allows the bats to strike with precision and rest between hunts, much like large carnivores. Big meals for small hunters. The bats often consumed prey nearly their own size, including frogs weighing up to 20 grams. One bat was recorded chewing for 84 minutes, indicating a substantial meal. On average, prey weighed 7% of the bat's body weight, equivalent to a human eating a 5 kilograms meal. Older bats were better at handling large prey, suggesting that hunting skills improve with experience. These bats also remember frog calls for years and learn from observing others. Rethinking predator ecology. This discovery challenges the traditional view that small predators must constantly hunt small, abundant prey due to their high metabolic rates. Instead, fringe-lipped bats show that precision, patience, and sensory adaptation can allow even tiny animals to hunt large, energy-rich prey efficiently. Lead author Leonie Bayer described them as big predators trapped in tiny bodies, while senior author Laura Stidsholt emphasized how biologging allowed researchers to experience the forest through the bat's ears. Genetically modified fungus offers new hope against malaria mosquitoes. Scientists have developed a novel method to combat malaria-carrying mosquitoes by using a genetically engineered fungus that specifically targets the disease vectors. According to a new study published this week, the approach demonstrates the potential of biotechnology to curb the spread of this deadly infection. The research team utilized the metarhizium fungus, which naturally infects insects, and enhanced its genome to produce toxins lethal to malaria mosquitoes but harmless to other insects and humans. In controlled laboratory tests, the modified fungus drastically reduced mosquito populations. Researchers emphasize that this method could become a more effective alternative to chemical insecticides, to which mosquitoes quickly develop resistance. Moreover, the biological approach lessens environmental harm. Field trials and long-term ecological studies are still needed to assess the safety and effectiveness of releasing this fungus into natural environments. Nonetheless, the findings offer hope for a new weapon against malaria, which kills hundreds of thousands of people each year. Experts highlight that combining genetic engineering with environmentally friendly biological methods could mark the beginning of a new era in controlling mosquitoes and other disease vectors.